Namaste students, I am JV Dr. Aziz Mohammad Khan from Department of Food and Biotechnology, Faculty of Agriculture and Veterinary Science, Jyoti Vidyapeeth Women's University, Jaipur. Today I am going to discuss about the alternate pathway of complement system. So what is the alternate pathway and what is the complement system, which we are going to discuss here. Complement system complement system is a system by which an antigen and antibody will bind to each other, and the complement protein will also bind to that protein and make a membranatic complex for the formation of a pore on the bacterial membrane surface, which will lead to the killing of the bacteria cell. So today I'm going to discuss about the alternate pathway. Alternate pathway. What is alternate pathway? It is the pathway which is also known as the properdin pathway, which because it a specific protein which is known as properdin P. Protein P is involved in this. Uh, it is the part of the innate immunity, and it is dependent on the antigen antibody complex formation. It is antibody dependent system, and the alternate pathway is slower than the classical pathway. If classical pathway do not go to the way, then alternate pathway will take place of the classical pathway, but it is slower than the uh, classical pathway. Uh, Same type of molecules, same type of proteins, such as C3, C4, C3, C5, C6, C7, C8, C9 are involved, which are being involved in the classical pathway. But here C1 is not involved, C2 is not involved, and C4 is not involved. The pathway starts from the C3. So that's why it is known as the alternate pathway. The pathway complement system goes in an alternative way. So that's why it is known as the alternate pathway. And a specific protein which is known as the protein is involved in the in this pathway. So how the alternate pathway is activated? C3 is contains some unstable thiazide bond which need to be bound, and this unstable bond makes C3 subject to slow spontaneous hydrolysis of the C3 into the C3A and C3B. The C3B is able to bind to the foreign surface antigen, and mammal cells contain salic acid, which activates C3B molecule. The C3 is converted into C3A and C3B. C3B is involved in the binding of the uh, mm, complement system, and C3A will be involved in the anahylatic reactions. Some another proteins are also involved in the alternate pathway, which were not in the classical pathway. The first one is factor B. Okay, factor B, C3B on the surface of a foreign cell binds to another plasma protein, which is known as the factor B. And this binding of factor B results in the exposure of a binding site of the another enzyme, which is called the factor D. So two factors are involved in this alternate pathway: factor B and factor D. First, factor B will bind, and it will facilitate the binding of the factor D. The factor D, the binding of the factor B to the C3B allows a protein enzymes called factor D to cleave factor B to the BB and BA. So First, C3 will cleave into the C3A and C3B. After that, C, uh, C3B will bind to the C uh, factor B. Okay. After that, factor B will facilitate the binding of the factor D, and when the factor D will bind, it will cleave the C3B molecule into the C3B and uh, and BB and BA. Two molecules will be separated. Factor BB will remain bound to the C3B while the factor BA and factor D will disperse. The complex C3B BB, okay, C3B BB is C3 convertase of the alternate pathway. So here we can share the screen. So this factor D, first of all, this factor B will bind to the C3B molecule, and after that, C3B molecule will bind to factor B and factor D. This factor D will facilitate uh, the cleavage of the factor B into the BB and the BA. The factor D and the factor BA will be dispersed away, and the remaining C3B and BB will be the pathway which will bound to the alternate pathway. This is the C3B, B, C3B, and C3B will bind, factor B will bind to the C3B. This is the factor D, and it will bind to the factor B, and the factor D will be removed, and BB and BB will be remaining bound to the C3B, but BE will be removed. After that, some one new molecule of the C3 will come, and it will be cleaved into the C3A and the C3B. C3A and C3B. The C3B will be removed. Sorry, the C3A will be removed, and the C3B will bind to another molecule which is known as factor B, and this factor B will facilitate the binding of the factor D, and after that this factor D will facilitate this factor will cleave the B factor C3B into the BBA and BBB. BBA and the factor D will be removed, and factor PB will still remain attached to the surface to the C3B. So this happens all around the microbial surface. Okay, it will be all around the microbial surface at different areas like this. Properdin factor BP is another molecule which will come. Properdin stabilize C3 convertase into the C3B BB on a microbial surface. So this C3B BB will be stabilized by the properdin factor, and may cleave over millions of C3 molecules also. It protects C3B from an inactivation by complement control mechanism. So after that, after binding of the C3 and activation of the C3 molecule, C5 will come, and it will bind to the complex which is C5, C5, C3B, C3B, and BB. Okay, C3B, C3B, C5, BP, and it will facilitate the cleavage of the C5 into C5b and C5a, which is conducted by the C3b BP complex, which is also known as the C5 convertage, and C5b will bind to this complex, and that after that C6 will come to the C5b. After that C7 will come and bind to the C6, C5, C6, C5b, and C7 will bind to the C8 complex, and after that uh, they will bind attach tightly on the surface of the microbe and form a membrane attack complex. After that formation of a membrane attack complex, C9 will come and it will bind to this membrane attack complex, and it will bind all around the microbial surface and making various kinds of pores on the surface of the microorganism. So each C9 will make a pore, and this completely make a whole pore on the microbial surface, which results in the destruction of the microorganism. So, so this is all about the alternate pathway. So what are the significance and what students will learn about the alternate pathway? So students will be able to understand what is the alternate pathway, what are the proteins which are involved in the alternate pathway, what is the another name of alternate pathway, uh, proteins such as C3b, C3bb, and C5, C6, C7, how will they attach to the membrane, and how this membrane attack complex will make pore on the surface of the microorganism, and how they will kill the microorganism. So student will be able to understand all these uh, phenomena. Now, uh, which kind of questions may arise? The first thing is that the questions may be based on the subjective questions or objective questions. The subjective questions may be asked such as the explain alternate pathway, explain the proteins which are involved in the alternate pathway, explain the mechanism of um, membrane attack complex formation in the alternate pathway, explain the cleavage of the C3b complex in the alternate pathway. So these questions can be asked in the examinations. So my lecture is over here. Thank you very much.